Welcome to another deep dive. Today we're plunging into a foundational topic that underpins so much of what makes us, well, us.
hmm
We're talking about amino acids, proteins, and enzymes, pulling insights from chemistry. An introduction to general organic and biological chemistry. Really trying to understand their relevance to health and life sciences.
It's a crucial deep dive because these aren't just, abstract chemical concepts.
Right.
Understanding these molecules is key to everything from how your muscles move to why a single tiny change can lead to something serious like sickle cell anemia.
Wow.
So our mission today is really to uncover the surprising why it matters behind these essential building blocks of life.
Exactly. Think of it as a shortcut to truly grasping the intricate chemistry powering your body, hopefully filled with those satisfying aha moments. So let's unpack this. When we talk about the chemistry of life, proteins often feel like the starting point. They're literally first in importance, aren't they?
They absolutely are. I mean, the name protein itself comes from the Greek word proteos, meaning first, which, well, it really highlights their critical and diverse roles.
Yeah.
Proteins are truly the workhorses of living organisms. They're just everywhere, from giving structure to your hair and nails, facilitating muscle movement, transporting vital molecules, acting as hormones,
And enzymes you mentioned.
and of course functioning as enzymes that catalyze pretty much every reaction in your body. It's the sheer versatility that makes them so fundamental.
That's an incredible range of functions. But okay, how are these complex, versatile molecules actually built? Where do they come from?
So proteins are constructed from smaller units called amino acids.
Okay.
There are 20 common amino acids that combine to form human proteins. And while they all share a common structural backbone, each one is uniquely defined by its side chain, known as the R group.
The R group. So that's what gives each amino acid its distinct personality, you could almost say.
Precisely. And that personality is critical because it dictates how the amino acid behaves chemically.
Right.
We classify amino acids based on their R groups. Some are nonpolar, meaning they tend to avoid water. They're hydrophobic, like valine, for instance.
Okay, water-fearing.
Others are polar, which means they readily interact with water. They're hydrophilic or water-loving.
Got it.
And within that polar group, you have those that are neutral or acidic or basic, depending on their R group's specific chemical properties.
And why does this classification, this non-polar-polar thing, matter so much for our bodies?
It's vital because the nature of these R groups directly impacts how proteins fold up into their specific 3D shapes
The fold.
and how they interact with their environment, like water inside our cells. That shape, in turn, dictates their function.
Okay.
Consider the stark example of sickle cell anemia. It's caused by the substitution of just one amino acid in the hemoglobin molecule, just one,
Just one. Out of how many?
out of hundreds. In this case, a nonpolar valine replaces a polar glutamate. This single tiny change profoundly alters the protein's behavior.
So
It leads to those crescent-shaped red blood cells that can't effectively carry oxygen, all from one R group swap.
that's amazing. It's such a small change with such devastating consequences. Speaking of essential, you hear about essential amino acids.
Yeah.
Our bodies can't make all of them, right?
That's correct. Of
Yeah.
the 20 common ones, our bodies can only synthesize 11. The other 9 are essential amino acids, meaning we must obtain them from our diet.
Okay.
This really underscores why a balanced diet isn't just, a good idea. It's a fundamental biological requirement.
So it's not just about getting enough protein overall, but the right combination of amino acids.
Exactly. Complete proteins, like those in most animal products, eggs, meat, fish, contain all 9 essential amino acids.
Okay.
Incomplete proteins, often found in plant sources like grains or beans, might be low or deficient in one or more.
So what do vegetarians or vegans do?
Well, here's a key insight. By combining complementary plant proteins, think rice and beans, a classic combo, where rice might be low in lysine but has other essentials, and beans are rich in lysine but maybe low in methionine, you can easily get all the essential amino acids. It's like a molecular synergy on your plate.
Clever. Okay, so once we have these individual amino acids, either from diet or synthesis, how do they actually link up to form these incredible protein chains?
They connect via peptide bonds. It's a specific type of amino bond. When they join, a water molecule is removed, and they form these chains.
Chains. Okay.
2 amino acids make a dipeptide, 3 a tripeptide. When you get really long chains, they're called polypeptides.
And this chain itself, it has a specific blueprint, I imagine, like a specific order.
Yes, absolutely. That's the primary structure of a protein. The unique, precise sequence of amino acids linked together by those peptide bonds. From one end, the N-terminus, to the other, the C-terminus.
And that sequence is critical.
It's absolutely fundamental. It determines everything else about the protein's final structure and function. Even if two peptides have the same amino acids, if the sequence is different, they're different proteins, likely with different functions.
Wow.
Think about hormones. The sequence GLU-his-pro stimulates the thyroid. Any other order of those same three won't work.
Incredible precision.
And determining this primary structure, like Frederick Sanger did for insulin back in 1953, was a massive scientific achievement.
So the sequence is the starting code, but surely it doesn't just stay a long, floppy chain floating around.
Not at all. That chain immediately begins to fold into distinct, stable shapes. This starts with the secondary structure.
Secondary, okay.
This level involves hydrogen bonds forming within the polypeptide backbone itself, not involving the R groups yet, just the backbone atoms.
And what shapes does that create?
The two most common forms are the coiled alpha helix. Think of it like a spiral staircase or a spring.
Right.
And the flatter, more extended beta-pleated sheet, which can be very strong, like in silk.
Okay, helix and sheet. Now, I've heard about proteins misfolding and causing diseases like Alzheimer's. Is that related to these secondary structures?
It absolutely is, and it's a critical link to health. In Alzheimer's disease, for example, there's a protein called beta amyloid. Normally, it seems to exist in an alpha helical form.
Okay.
But in Alzheimer's patients, these proteins undergo a change, adopting a sticky beta-pleated sheet structure instead.
Uh-oh.
Right. These beta sheets then tend to clump together, aggregating into insoluble plaques in the brain. These plaques damage neurons and interfere with nerve signals. So yes, that shift in secondary structure is a major part of the disease's pathology.
That's a really powerful example of how structure dictates function, or dysfunction in this case. What about the next level of complexity, after secondary?
That takes us to the tertiary structure. This is the overall complex three-dimensional shapes that the entire polypeptide chain folds into.
The whole thing.
The whole thing. And this folding is driven primarily by interactions between the R groups of the amino acids along the chain.
Ah, so now the R groups come into play.
Exactly. It's a very specific, unique, and often compact shape, stabilized by a whole bunch of interactions.
Like what?
Well, you have hydrophobic interactions, the nonpolar R groups trying to hide from water inside the protein. Then hydrophilic interactions, the polar R groups wanting to be on the outside, interacting with water.
Makes sense.
You also get salt bridges, which are ionic bonds between charged acidic and basic R groups, plus hydrogen bonds between polar R groups. And, importantly, strong covalent disulfide bonds that can form between two cysteine amino acids.
So the R groups are essentially playing a molecular game of like hide and seek with water, while also forming specific links and attractions to hold the final 3D shape together.
That's a great way to put it. And this specific 3D tertiary shape is absolutely crucial for the protein's biological activity.
Can you give an example?
Think of collagen, the main protein in our connective tissue. It actually forms a triple helix 3 polypeptide chains wound around each other like a rope. This structure gives it immense strength. And it's why vitamin C is so important. It's needed by enzymes that help form extra stabilizing bonds within the collagen structure. Lack of vitamin C weakens collagen, leading to problems like scurvy.
Right, I remember that link. Okay, so primary, secondary, tertiary. Is there more? What if proteins need even more complexity?
There is. That's the quaternary structure. Many proteins are fully functional as a single polypeptide chain folded into its tertiary structure. But others require 2 or more of these folded polypeptide chains or subunits to assemble together to become active.
Like a team.
Exactly like a team. Hemoglobin, the protein that carries oxygen in our blood, is the classic example. It's composed of 4 separate polypeptide chains, 2 alpha chains and 2 beta chains.
4 subunits.
4 subunits. Each one has a heme group that binds oxygen, but all 4 must be assembled correctly in the quaternary structure for hemoglobin to transport oxygen efficiently throughout your body.
And the interactions holding these subunits together, are they similar to the ones in tertiary structures, like the R group interactions?
Precisely the same types of interactions. Hydrogen bonds, salt bridges, hydrophobic interactions, sometimes disulfide bonds, but now occurring between different polypeptide subunits.
Okay, that ties it together. And this brings us back to sickle cell anemia again, doesn't it?
It does, perfectly. Remember that single amino acid change in the beta chain? Replacing the polar glutamate with a nonpolar valine.
Yeah.
That change happens on the surface of the beta subunit. Because valine is nonpolar, it creates a sticky patch. This sticky patch attracts other nonpolar regions on other hemoglobin molecules, especially when oxygen levels are low.
So they clump.
They polymerize. They stick together, forming long, rigid, insoluble fibers inside the red blood cell. This distorts the cell into that characteristic sickle shape.
And that causes the blockages and pain.
Yeah, exactly. Blocked blood flow, lack of oxygen delivery, intense pain, all stemming from that one change affecting quaternary structure interactions. It's a powerful illustration of how critical these structures are.
It truly is a delicate balance. So what happens if these precise structures, secondary, tertiary, quaternary, get messed up or disrupted?
That's called denaturation. It's when a protein loses its specific organized 3D shape. Its secondary, tertiary, or quaternary structure gets disrupted.
Does it break the chain itself, the primary structure?
Usually not. The peptide bonds holding the amino acid sequence together, the primary structure, typically remain intact. But the intricate folding is lost. The protein essentially unfolds, often becoming tangled and non-functional, like a loose piece of cooked spaghetti compared to its original precise shape.
And what triggers denaturation? What can cause a protein to unfold like that?
Several factors can do it. Heat is a major one. Temperatures above, say, 50 degrees C or 122 degrees F provide enough energy to disrupt those weaker interactions like hydrogen bonds and hydrophobic interactions.
Like cooking an egg.
Exactly like cooking an egg. The heat denatures the albumin proteins, turning the clear egg white into a solid white mass. This is also why high fevers can be so dangerous. They can start to denature crucial enzymes in your body.
Okay, heat. What else?
Acids and bases, so extreme changes in pH. These disrupt hydrogen bonds and, importantly, the salt bridges that rely on specific charges on acidic and basic R groups. Think about how adding acid, like lemon juice or vinegar, curdles milk to make cheese or yogurt. That's denaturation of milk proteins.
Right. Any others?
Yes. Certain organic compounds, like rubbing alcohol, can disrupt the hydrophobic interactions that hold nonpolar groups together. That's why alcohol swabs are used as disinfectants. They denature bacterial proteins. Heavy metal ions like lead, mercury, silver can react with sulfur atoms and cysteine R groups, disrupting disulfide bonds or form bonds that mess up salt bridges.
Nasty stuff.
Very. And even physical agitation, like whipping egg whites vigorously.
Yeah.
The mechanical force can stretch the polypeptide chains enough to break the stabilizing interactions.
Okay, so lots of ways to mess up protein structure. Now, we've touched on enzymes a few times as a type of protein, but they seem so important they probably deserve their own spotlight, don't they?
Oh, absolutely. Enzymes are phenomenal biological catalysts.
Yeah.
They are essential for life as we know it.
Catalysts, meaning they speed up reactions.
Dramatically.
Yeah.
Most chemical reactions needed for life, digestion, energy production, DNA replication would happen far too slowly at body temperature and pH without enzymes. Or they might require conditions incompatible with life. Enzymes make these reactions happen fast enough for survival.
How fast are we talking?
It varies, but some are incredibly efficient. Take carbonic anhydrase, an enzyme in your red blood cells. It helps convert carbon dioxide and water into bicarbonate and hydrogen ions, so CO2 can be transported from tissues to the lungs. It can process about 1 million substrate molecules per second.
1 million per second? That's hard to even imagine. So how do they achieve that speed boost?
They work by lowering the activation energy of a reaction. Think of activation energy as the energy hill that reactants, substrates need to climb before they can turn into products. Enzymes provide an alternative reaction pathway with a much lower hill. They stabilize the transition state, making it easier and faster for the reaction to occur.
Got it. Lowering the barrier. And how are these speed demons named? Is there a system?
Generally, yes. Enzyme names typically describe the reaction they catalyze and end with the suffix ase.
Gaze, like
sucrose, which catalyzes the hydrolysis of sucrose, or lipase, which hydrolyzes lipids, and oxidase carries out oxidation. It's usually quite descriptive.
Simple enough. Now, where does the actual reaction, the catalysis, happen on the enzyme molecule?
Each enzyme has a unique, complex, three-dimensional shape.
All right.
And within that structure, there's a specific region called the active site.
The active site.
Yes, it's often a small pocket or cleft on the enzyme surface. This active site has a shape and chemical environment that is precisely complementary to the enzyme's specific reactant molecule, which we call the substrate.
So it fits perfectly.
It binds the substrate specifically. The R groups of certain amino acids lining the active site interact with the substrate through various temporary bonds, hydrogen bonds, ionic interactions, etc. These interactions hold the substrate in the optimal position for the reaction to occur.
So it was once thought of as a simple lock and key mechanism, right? The enzyme is locked, the substrate is the key. How has that idea evolved?
That lock and key model was a useful early concept, emphasizing the specificity only the right key fits the lock.
Hmm.
However, we now understand it's a bit more dynamic. The currently accepted model is the induced fit model.
Induced fit.
Yeah, this model recognizes that the active site isn't perfectly rigid before the substrate binds. Instead, the binding of the substrate can actually induce a slight change in the shape of the active site and maybe even the substrate itself.
Ah, like a handshake.
Exactly. It's more like a handshake adapting for the best grip rather than a rigid key fitting a rigid lock. This interaction achieves the optimal alignment for catalysis.
That makes a lot more sense, actually. A flexible interaction. And once the reaction happens, the products are formed. What happens to the enzyme itself?
The enzyme releases the products, and then, importantly, the enzyme returns to its original shape, ready to bind another substrate molecule and catalyze the reaction again.
So it's reusable.
Highly reusable. It's not consumed or permanently changed in the process, which is why a small amount of enzyme can process a large amount of substrate over time.
Okay. Now, you mentioned earlier, way back, that enzymes can be used as diagnostic tools in medicine. How does that work?
Ah, yes, that involves isoenzymes, sometimes called isozymes.
Lysoenzymes.
These are slightly different forms of the same enzyme that catalyze the exact same reaction, but they are produced in different tissues or organs within the body.
Different versions for different places?
Pretty much. They
Okay.
often have small variations in their amino acid sequence,
Yeah.
usually in the quaternary structure subunits, which don't affect the active site much but give them slightly different properties.
So how does that help doctors? If a specific isoenzyme shows up in my blood, what does it tell them?
Exactly. Normally, enzymes stay inside their respective cells, but when there's damage or disease in a specific organ, like the heart or liver, the cells in that organ may die and rupture, releasing their contents, including their characteristic isoenzymes, into the bloodstream.
Ah, leakage.
Precisely. So by measuring the levels of specific isoenzymes in a patient's blood serum, doctors can get clues about which organ might be damaged.
Can you give an example?
Sure. After a heart attack, a myocardial infarction, heart muscle cells die and release their specific isoenzymes. Levels of creatine kinase isoenzyme CKMB, or CK2, and lactate dehydrogenase isoenzyme LDH1, which are abundant in heart muscle, become elevated in the blood. Measuring these helps confirm the diagnosis and assess the extent of damage.
Fascinating. So enzymes are clearly essential, powerful, but are they always working at full throttle? What can affect their activity, how fast they work?
No, definitely not always full throttle. Enzyme activity, or the rate at which they catalyze reactions, is incredibly sensitive to the reaction conditions. Three major factors are temperature, pH, and the presence of specific molecules called inhibitors.
Okay, let's take temperature first. I imagine there's an ideal range, like with body temperature.
Yes, exactly. Enzymes have an optimum temperature at which they show maximum activity. For most human enzymes, this is around 37 degrees C, normal body temperature.
And what happens if it's too cold or too hot?
At lower temperatures, activity slows down simply because molecules move slower, leading to fewer collisions between enzyme and substrate. But if the temperature goes too high, typically above 50 degrees C or so for human enzymes, the heat energy starts to disrupt the weak bonds holding the enzyme's tertiary and quaternary structure.
The denaturation again.
Denaturation again. The enzyme loses its specific 3D shape, including the active site, and its catalytic activity is lost, often permanently. This is why very high fevers can be so dangerous.
So heat effectively breaks the enzyme's functional shape. What about pH? Does that work similarly?
Very similarly, enzymes also have an optimum pH range where they function best. This pH helps maintain the proper ionization states of the acidic and basic R groups, crucial for both the enzyme's tertiary structure and the interactions within the active site.
And for most enzymes in our body, that's around neutral pH.
For most, yes, around the physiological pH of about 7.4. If the pH becomes significantly too acidic or too basic, it changes the charges on those crucial R groups.
How does that affect it?
It disrupts salt bridges and hydrogen bonds, altering the tertiary structure and the shape of the active site, leading to a loss of activity. Denaturation due to pH changes.
But aren't there exceptions? Like in the stomach?
Absolutely. Digestive enzymes are great examples of adaptation. Pepsin, the main protein digesting enzyme in the stomach, has an optimum pH of around 1.5 to 2.0.
Wow, very acidic.
It has to be, to function in a highly acidic environment of the stomach. Its activity drops dramatically if the pH rises towards neutral. So optimum pH is specific to the enzyme and its environment.
Okay, temperature and pH. What about the third factor, inhibitors? They sound like they stop enzymes from working.
That's precisely what they do. Inhibitors are molecules that bind to an enzyme and cause it to lose or reduce its catalytic activity. They essentially interfere with the enzyme's ability to bind its substrate or carry out the reaction.
Are there different ways they can inhibit?
Yes, there are a few key types. First, you have competitive inhibitors.
Competitive.
These inhibitors often have a chemical structure and shape that's very similar to the enzyme's normal substrate. They literally compete with the substrate for binding to the active site.
So if the inhibitor gets there first, the substrate can't bind.
Exactly. If the inhibitor is occupying the active site, the substrate is blocked and no reaction occurs. The good news here is that this inhibition is often reversible. If you increase the concentration of the actual substrate, it can eventually outcompete the inhibitor for the active site, restoring enzyme activity.
Okay, makes sense. What's another type?
Then there are non-competitive inhibitors. These molecules don't resemble the substrate, and they don't bind to the active site itself.
So where do they bind?
They bind to a different site on the enzyme molecule. But this binding causes a change in the enzyme's overall three-dimensional shape.
Ah, a conformational change.
Right. And this change distorts the active site, so even if the substrate can still bind, it might not fit properly, or the enzyme can't perform the catalysis effectively.
And can you overcome this by adding more substrate?
No, because the inhibitor isn't competing for the same site. Adding more substrate won't reverse the effect of a non-competitive inhibitor. Heavy metal ions, like lead or mercury, often act as non-competitive inhibitors by binding to R groups away from the active site.
I see. Is there a third type?
Yes, the most drastic kind.
Mm.
Irreversible inhibitors. These are often highly toxic substances.
How do they work?
They typically form a strong, permanent covalent bond with an amino acid side group, usually one that's critical within the active site itself. This permanently inactivates the enzyme, essentially destroying its catalytic function forever.
So things like certain poisons, maybe some insecticides or nerve gases would fall into this irreversible category.
Exactly right. Many potent insecticides and nerve gases work by irreversibly inhibiting crucial enzymes involved in nerve signal transmission, like acetylcholinesterase.
And antibiotics do some work this way.
Yes, some do. Penicillin, for example, is an irreversible inhibitor of an enzyme called transpeptidase, which bacteria need to build their cell walls. By permanently blocking this enzyme, penicillin prevents the bacteria from forming strong walls, eventually causing them to rupture and die. Crucially, human cells don't have this enzyme, which is why penicillin is selectively toxic to bacteria.
That's incredibly clever, using inhibitors as targeted weapons.
It's a cornerstone of pharmacology and medicine. Understanding enzyme inhibition allows us to design drugs that specifically target enzymes involved in disease processes or in pathogens.
Wow, that was a truly deep dive into the molecular machinery of life. From the simplest amino acid building block, through the complex dance of protein folding, to the precise and vital actions of enzymes, it's just so clear how foundational these concepts are.
It really is. We've seen how, a single amino acid change can lead to a devastating disease like sickle cell anemia, how protein misfolding plays a role in complex conditions like Alzheimer's, and how enzymes are these incredibly finely tuned biological catalysts essential for our very survival. Understanding these structures, their functions, and their delicate balance is truly at the heart of health, disease, and medical science.
It really makes you think about how incredibly interconnected everything is, right down to the molecular level. And this knowledge isn't just for, well, textbooks or exams. It's the absolute basis for understanding disease, for developing effective treatments, and even designing new medications that target these precise molecular interactions.
Absolutely. And it raises an important, perhaps even profound question. What other seemingly small molecular details, maybe ones we haven't even discovered yet, might have equally profound and unexpected impacts on our health, our biology, and the world around us? It feels like there's still so much more to explore in these fundamental building blocks.
A fascinating thought to end on. And that's all the time we have for this deep dive. We really hope you've enjoyed uncovering these fascinating insights with us today. Until next time, keep that curiosity alive.